For over 10,000 years, the howl of a dire wolf hadn't echoed across the earth. The species was thought to be lost forever, extinct beyond recovery. But that might be about to change. Colossal Biosciences, a company specializing in de-extinction, has taken on the extraordinary mission of bringing the dire wolf back using advanced genetic engineering. And in March of this year, the world saw something incredible. 6-month-old puppies that looked astonishingly like dire wolves. Ben Lamb, the company's founder, explained, "We need new tools for conservation. We want to create value, inspire people, and intelligently resurrect species that can benefit ecosystems." To achieve this, scientists had to first understand dire wolves at the genetic level. They extracted DNA from 2 fossilized bones, one around 13,000 years old, the other roughly 72,000. Using these samples, the researchers reconstructed 2 dire wolf genomes and compared them to the genomes of modern wolves. They searched for genetic differences, the key traits that made dire wolves unique. After identifying them, the team made 20 targeted edits across 14 genes in the gray wolf's DNA. This wasn't a full resurrection of the species, but rather the creation of a genetically modified gray wolf with unmistakable dire wolf features: white fur, larger body size, and that haunting howl you heard at the beginning. So how was this done? Scientists removed the original DNA from wolf eggs and replaced it with the edited genetic material. These modified cells developed into embryos, which were then implanted into large mixed-breed dogs to carry the pups. On the 1st of October 2024, 2 were born, Romulus and Remus. In January, their younger sister Calesia joined them. Each is expected to grow up to 2 meters in length and weigh around 70 kilograms. And the dire wolf is just the beginning. Colossal's de-extinction list also includes the Mauritian dodo and the Tasmanian tiger. Most ambitious of all is the plan to resurrect the woolly mammoth by 2028, a project far more complex than the dire wolf. The mammoth genome differs from that of modern elephants by millions of genetic variations. Thousands of precise edits will be needed to create a cold-resistant elephant capable of thriving in Arctic environments. And with elephants having a 22-month pregnancy, the longest gestation period of any mammal, progress is slow. Yet the team remains optimistic. They've already created cells with mammoth genes that produce cold-resistant hair and fat. The next step is to transfer these changes into an elephant egg to create the first mammoth-like embryo in over 4,000 years. Experts warn that by 2050, Earth could lose up to 30% of its genetic diversity. Genetic engineering may help halt that decline. The Colossal team believes that resurrecting extinct species can also protect those still living. Technologies developed for mammoths and wolves are already helping endangered animals today. For instance, breakthroughs from the Dire Wolf Project have been used to clone 4 red wolves and coyotes, a major step toward restoring genetic diversity to the endangered red wolf population. Beth Shapiro, evolutionary biologist and Colossal's scientific director, put it best. "It would be amazing to see a mammoth. It would be great to bring a keystone predator back to Tasmania's ecosystems. But the tools we're developing have immediate applications for species that haven't yet gone extinct." The problem of genetic bottlenecks is well known to scientists. When a population becomes too small, animals begin to breed with close relatives. Genetic diversity declines, harmful mutations accumulate, and the entire species becomes more vulnerable to disease. Humanity has faced a similar threat. DNA analysis shows that around 70,000 years ago, our ancestors nearly went extinct. Perhaps it was caused by the eruption of the Toba supervolcano in Indonesia, which led to a sharp global cooling. Whatever the cause, the human population may have dropped to just a few thousand individuals. All modern humans are descendants of this tiny group of survivors, which is why we are so genetically similar. The difference between any 2 humans is only about 0.1% of their genome. By comparison, chimpanzees from neighboring groups can differ 10 times more. Thanks to modern technology, scientists can now study not only our direct ancestors, but also our long-lost cousins. In 2010, researchers sequenced the Neanderthal genome for the first time using bones that were 40,000 years old. It turned out that Neanderthals didn't simply disappear. They interbred with our ancestors. Today, every person outside of Africa carries between 1% and 3% Neanderthal DNA. These ancient genes still influence us. Some help our immune systems fight infection, while others slightly increase the risk of diabetes or depression. Then, in 2008, archaeologists discovered a tiny piece of finger bone in Denisova Cave in the Altai Mountains. When scientists sequenced its DNA, they were stunned. It belonged to an entirely new species of ancient human, the Denisovans. Only a few teeth and bone fragments remain, but their genetic legacy lives on in modern populations of Asia and Oceania. The people of Papua New Guinea and Melanesia, for example, carry up to 6% Denisovan genes. From them, Tibetans inherited a remarkable adaptation, a gene that allows their blood to carry oxygen efficiently at high altitudes. These findings revealed that human history is far more complex than once believed. Our ancestors didn't simply replace other human species, they merged with them, exchanging genes and evolutionary advantages. We owe this knowledge to a revolution in our understanding of the code of life. In 1953, James Watson and Francis Crick discovered the double helix structure of DNA. But reading the entire human genetic code proved far more difficult. In 1990, the Human Genome Project began one of the most ambitious scientific endeavors in history. Thousands of scientists from 6 countries worked for 13 years, using room-sized sequencing machines and spending over $3 billion. Finally, in 2003, they announced success. The complete human genome had been decoded, roughly 6 billion letters of genetic code. The results were surprising. Scientists had expected to find around 100,000 genes, but there were only 20 to 25,000, roughly the same as a microscopic worm. Even more surprising, only 1% of our DNA actually codes for proteins. The remaining 99%, once dismissed as junk DNA, turned out to be an intricate control system that determines when and where genes are activated. About 8% of the human genome consists of fragments of ancient retroviruses, remnants of infections that occurred millions of years ago. Astonishingly, one of these viral genes now plays a crucial role in forming the placenta. In other words, without this junk DNA, none of us would be born. Another remarkable fact: all humans are 99.9% genetically identical. We differ by just 1 letter in 1,000. And yet, that tiny difference defines everything: eye color, height, disease risk, even aspects of personality. Geneticists have identified genes linked to intelligence, longevity, and athletic ability. They've learned why some people can drink alcohol without issue, while others turn red after a single sip. They've uncovered the origins of hereditary diseases and how to fix them. And it turns out, we are more than just human at all. Our bodies host trillions of bacteria, viruses, and other microorganisms collectively known as the microbiome. It weighs around 2 kilograms and contains 100 times more genes than our own DNA. This vast microbial network affects our mood, our immunity, and even our metabolism. It can influence whether we gain weight or stay lean. We are not single organisms, but living ecosystems, a fusion of human and microbial genes working together. This understanding is already transforming medicine. Doctors now tailor treatments to a patient's genetic profile. They design personalized drugs that target specific genetic variants. And in some cases, they even edit genes directly inside the body, repairing inherited defects at the source. Here's one example. Children born with Leber's congenital amaurosis carry a single extra letter in one of their genes, a tiny typo that causes blindness. For years, scientists searched for a way to help them. Finally, they succeeded. They developed a treatment called EDIT-101, a harmless virus carrying molecular scissors inside. The virus is injected directly into the eye, where it finds the faulty gene and precisely cuts out the extra letter. The results were astonishing. After just 1 year, 11 out of 14 patients showed improved vision. Some could recognize letters on a screen for the first time. Others saw traffic lights, colors they'd never seen before. And remarkably, no serious side effects were reported. Another breakthrough uses a different approach treatment outside the body. In the case of sickle cell anemia, doctors remove stem cells from the patient's bone marrow. In the lab, these cells are edited with molecular scissors that repair the defective gene. Then, the corrected cells are infused back into the body. This therapy was officially approved in 2023, and the results were extraordinary. 93% of patients experienced complete relief from painful crises. Their blood returned to normal, just like in healthy individuals. A single-dose cure sounds almost like science fiction, yet it's already reality. A gene-editing drug is injected into a vein, travels to the target organ, and switches off the malfunctioning gene. The concentration of the harmful protein drops by 90%, and the effect lasts for more than a year after only one treatment. Why is this possible now? 3 major breakthroughs made it real. First, genome sequencing has become incredibly cheap. What once cost millions of dollars can now be done in just a few days for about $200. Second, scientists have mastered precise delivery, directing drugs exactly where they're needed, to the liver, to the eyes, or to the bone marrow, while leaving healthy tissues untouched. Third, medicine has become personalized. Doctors can now analyze a patient's genome to determine exactly which treatment and dosage will work best for them. Clinical trials are already underway for hemophilia and muscular dystrophy, and dozens of new gene therapies for rare diseases are on the horizon. If successful, thousands of once incurable conditions could soon be treated or even cured. Not long ago, a genetic disease was a life sentence. Today, it's just a bug in the code, something we can fix. Our genes no longer define our destiny. They've become instructions, and medicine has learned how to edit them. And all of this has been made possible thanks to an unlikely ally: artificial intelligence. It's AI that analyzes the genomes of millions of people and finds hidden links between genes and disease. It's AI that predicts which cells to edit and how. It's AI that simulates millions of possible treatments, selecting the one that's most effective. No human brain could process such vast data, but a machine can, in seconds. And pharmaceutical companies are already using this technology to design the next generation of medicine. For example, AstraZeneca is already using artificial intelligence in medicine. Their system, called MILTON, works like a digital oracle. It analyzes 67 medical indicators and can predict the risk of developing more than 1,000 different diseases. MILTON was trained on the data of 500,000 participants from the UK Biobank. Today, it can look at your blood tests, blood pressure, and other biomarkers and estimate your risk of developing dementia 10 years before symptoms appear. This isn't guesswork. The algorithm finds hidden patterns in data that the human eye simply cannot detect: tiny fluctuations in blood chemistry, subtle deviations in blood pressure, invisible trends that together form a map of future illness. Another milestone for AstraZeneca came through its collaboration with benevolent AI. Their artificial intelligence independently discovered a new biological target for treating chronic kidney disease without any human prompting. The system analyzed billions of connections between genes, proteins, and research papers, building hypotheses on which proteins might be blocked to treat the disease. When biologists tested the AI's hypothesis in the lab, it was correct. This became one of the first cases in history where an AI-generated target led to a real drug. In the past 5 years, we've seen more breakthroughs in genetic medicine than in the previous 50. And this is only the beginning. Today, we can correct a single gene. Tomorrow, we'll be able to correct dozens. The day after tomorrow, we may prevent diseases before birth. And in the coming decades, we might even learn how to defeat aging itself. But to understand where this road leads, we must return to the very foundation of life, to the microscopic protein machines that carry out the code of existence. Through its This Is Science initiative, AstraZeneca promotes new discoveries in oncology, cardiology, immunology, and beyond, helping the public grasp the beauty and complexity of modern science. That same idea inspires me. In this video, you'll see how humans and machines are beginning to rewrite the rules of life itself, and why some scientists now believe that we could be the last generation to age and to die of disease. What is life? There's a simple yet profound definition. Life is based on complementary interactions between macromolecules. Let's break that down. Complementarity is like yin and yang, like a right hand and a left hand. Every process in our cells is a chemical reaction. Molecules fit together in a complementary way, interact, and transmit signals. Think of it like Lego or a puzzle. The pieces have to fit perfectly for meaning to appear. That's how life works. To understand the structure of life, we must understand the three-dimensional shapes of the particles that make it possible: proteins. We've become incredibly skilled at reading genomes. We can now decode DNA in hours, and for just a few $100. We understand the rules of the genetic code, how a DNA sequence translates into a sequence of amino acids. But that's where the real challenge begins. A protein is just a long chain of amino acids, like a strand of spaghetti. To function, it has to fold into an exact three-dimensional shape. Only then can it interact with other molecules and do its job. Understanding how a protein folds is one of the great arts and great mysteries of biology. The problem is, proteins are unimaginably small. Imagine if a carbon atom were the size of a human being. At that scale, a single protein would tower as high as the Spasskaya Tower of the Moscow Kremlin. And your DNA? It would stretch from the Earth to the Moon dozens of times over. Proteins can't simply be photographed under a microscope. For over 60 years, biologists painstakingly mapped the structures of about 180,000 proteins molecule by molecule. Then, in just a few years, a team of only 15 researchers determined the structures of 200 million proteins, virtually every one known to science. A protein isn't one specific molecule. It's a type of compound, like a string of beads, each bead representing one of 20 amino acids. These 20 building blocks can be combined almost infinitely. Proteins are the workers of your body. They digest your food, shape your face, determine how fast signals travel through your brain, copy your DNA, and even form your memories. There are around 20,000 different proteins inside you, each performing its own task with incredible precision. But proteins don't decide what to do by themselves. They're like microscopic robots following instructions. Those instructions are written in nucleic acids, DNA, and RNA. On their own, nucleic acids do almost nothing. They merely store information. And in that simple relationship lies the secret of life. Information determines function. The amino acid sequence determines what a protein can do, and the protein's shape determines its role in the symphony of the cell. Yet discovering the structure of even a single protein is an enormous challenge. In the 20th century, scientists invented a clever technique. They crystallized proteins and bombarded them with X-rays. By analyzing the diffraction patterns, they could infer the protein's shape. Biochemist John Kendrew spent 12 years deciphering the structure of a protein called myoglobin. At first, he used horse heart tissue, but it didn't contain enough of the protein. Then he had an idea. Diving mammals must have stronger, denser muscle proteins. He obtained a piece of whale meat and finally produced crystals that worked. When the structure was revealed, it shocked everyone. Scientists expected to see something clean and orderly, but instead, they found a chaotic, tangled form. A bizarre structure so complex that one researcher jokingly called it the turd of the century. It was nothing like the tidy diagrams of textbooks, but it was real. And for this discovery, in 1962, John Kendrew received the Nobel Prize. Over the next 20 years, only about 100 protein structures were deciphered. Even today, this process remains incredibly difficult and expensive. Determining the structure of a single protein can cost tens of thousands of dollars. Scientists began searching for an alternative. Sequencing the amino acid chain costs only about $100. If only they could predict the full three-dimensional structure directly from this sequence, they could save enormous time and money. Chemist Linus Pauling made one of the few accurate early predictions in biology. He proposed that amino acids can form helices and sheets, but progress stalled. Proteins turned out to be far more complex than anyone expected. Evolution hadn't created them from scratch. It had assembled them gradually over billions of years. Biologist Cyrus Leventhal calculated that even a short chain of just 35 amino acids could fold in an astronomical number of ways. A supercomputer trying every possibility would take 200 times longer than the universe has existed to find the right one. Still, Professor John Molt refused to give up. In 1994, he created a global scientific competition called CASP, the Critical Assessment of Structure Prediction. The challenge was simple in concept, but monumental in difficulty. Build a computer program capable of predicting a protein structure from its amino acid sequence. Each year, participants were given a protein whose structure was known only to the organizers. The predicted models were compared to experimental results. A perfect match earned 100 points. A score of 70% was considered good, and anything above 90% was exceptional. The first meetings were held in an old wooden chapel, and a curious ritual emerged. Whenever someone's prediction was far off, the audience would stomp their feet on the floor, and they stomped a lot. In the very first year, no team managed to score above 50 points. A few years later, biologist David Baker developed an algorithm called Rosetta. Baker found a way to accelerate the calculations by distributing them across thousands of personal computers in homes, schools, and libraries around the world. Volunteers installed the Rosetta at-home program, which ran as a screensaver, showing animated protein chains twisting and folding as their computers contributed to science. Some volunteers even wrote to the team, joking, "Judging by these pictures, we could probably do better ourselves." Baker took that literally and turned protein folding into a game. The result was Fold-It, a puzzle game where players manually folded virtual proteins into the shapes they thought were correct. For 10 days straight, more than 50,000 players worked on a single challenge, the enzyme of the human immunodeficiency virus. Their collective solution was compared to crystallography data, and it was correct. The Fold-It community was credited as co-authors on a peer-reviewed scientific paper. Among those players was an exceptional mind, Demis Hassabis, a former chess prodigy. Shortly before, he had founded the artificial intelligence company DeepMind, famous for its AlphaGo algorithm, which defeated world champion Lee Sedol. I discussed that story in detail in my video about the unsolvable problem in AI and why it matters. Hassabis never forgot his experience with Fold-It. "I was fascinated from a game design perspective," he later said. "How amazing would it be to teach a computer to think like those players, not professional scientists, but ordinary people who could intuit complex biological structures?" The DeepMind team organized a week-long hackathon to teach artificial intelligence how to play Fold-It. That event marked the birth of the AlphaFold project. The first version of AlphaFold was a fairly standard neural network, similar to those used in computer vision. It was trained on a vast database of known protein structures. The inputs were the amino acid sequences, along with information about evolutionary patterns. In evolution, natural selection works through random mutations in DNA. Species change, but their core proteins tend to stay remarkably similar. For example, hemoglobin looks nearly identical in humans, horses, and cats. Evolution seems to say, if it isn't broken, don't fix it. By comparing the same protein across multiple species, scientists can identify which regions are crucial for its structure. Even if the sequences differ, paired mutations can reveal which amino acids sit close together in three-dimensional space. This evolutionary insight became a cornerstone for AlphaFold's design. This data was then used by a secondary algorithm to fold the chain into a 3D configuration. With this approach, AlphaFold entered the CASP competition and achieved a remarkable breakthrough. After a few refinements, it scored around 70 points, a solid success, but DeepMind wanted more. To go further, they had to rethink everything. In AlphaFold 2, we changed the deep learning approach so that individual parts of the system developed a true understanding of proteins, their geometric, physical, and evolutionary principles. Explained John Jumper, lead author of AlphaFold at Google DeepMind. These principles had to be built into the core of the network, not just added on top. That gave us a huge boost in accuracy. The upgrade came through three major innovations. First, they unleashed more computing power. DeepMind had access to some of the most advanced resources in the world. Google's powerful tensor processing units built specifically for AI computation. Second, they ensured the model had access to a rich and diverse dataset. Yet Jumper emphasized that more data wasn't the key. "I think the importance of data quantity is overstated. AlphaFold 2 was trained on the same dataset as AlphaFold 1. The difference was in how it was trained. When your learning process becomes more efficient, the lack of extra data matters much less." Third, the DeepMind team completely redesigned the neural network architecture, introducing a new framework called EvoFormer. EvoFormer operates on a dual tower system. The first tower handles biological information, the evolutionary relationships between amino acids. The second tower focuses on geometry, the spatial distances and angles that define the protein structure. In the original AlphaFold, one tower had to predict the other. In AlphaFold 2, EvoFormer changed that entirely. Both towers worked simultaneously, connected by a continuous feedback bridge. Information flowed back and forth between them 48 times, refining the data with each pass until both towers converged on the same structural truth. Finally, the system assembled the full three-dimensional protein. For each amino acid, it identified 3 key atoms that define its orientation in space. And the most elegant part, the AI didn't know the sequence as a chain. It simply received a collection of parts and figured out on its own how they fit together. The chain emerged naturally as a consequence of geometry, not instruction. In December 2020, DeepMind returned to the CASP competition with AlphaFold 2, and this time they succeeded. The predicted protein structures were almost indistinguishable from the experimental ones. The system finally broke the 90-point barrier, something once thought impossible. Over 6 decades of X-ray crystallography, scientists around the world had determined roughly 180,000 protein structures. Then AlphaFold came along and revealed over 200 million, essentially mapping nearly every protein known to nature. In just a few months, it accelerated biological research by decades. Thanks to AlphaFold, scientists developed malaria vaccines, gained new tools to fight antibiotic resistance, and uncovered the molecular mechanisms behind diseases from schizophrenia to cancer. In 2024, John Jumper and Demis Hassabis were awarded half of the Nobel Prize in Chemistry for solving this 50-year-old mystery. The other half went to David Baker for pioneering the creation of entirely new proteins. Today, you can design a protein on a computer and have a physical molecule within days. Researchers are already using this technology to design synthetic antibodies against snake venom, targeted cancer-fighting proteins, and enzymes that absorb methane or break down plastic. And this is only the beginning. If AlphaFold can predict protein structures, the next frontier is to redesign them. Imagine taking any protein and modifying it to suit our needs, making an enzyme work dozens of times faster, or creating one that captures carbon dioxide directly from the air. Baker's team is working on this right now. They're designing proteins that have never existed in nature, changing just a few amino acids to unlock entirely new functions. One of their creations glows brighter than any natural fluorescent protein. Another binds to toxins and removes them from the body. A third acts like a molecular switch, turning on and off on command. The next step goes even further. Protein nanomachines. Scientists are assembling them from individual protein components like biological Lego. These nanoscale robots, just a few billionths of a meter in size, could one day deliver drugs directly to a target cell or repair damaged tissue molecule by molecule. Human testing is still years away, but DNA-based nanorobots are already being trialed in animals. These larger machines can identify cancer cells and destroy them from within, reducing tumor growth in mice by up to 70%. Conventional biochemistry acts like a bomb, striking the entire body. But a DNA nanorobot works like a sniper, hitting only its intended target. There's a problem, though. Designing a new protein on a computer is only half the battle. You still need a living cell to produce it. That means reprogramming bacteria or yeast to synthesize the molecule, essentially installing a new app on a biological smartphone. But instead of code, scientists upload genetic instructions for protein production. Sometimes the cells misbehave. The protein comes out deformed, or it doesn't work at all. When that happens, the researchers return to their computers, redesign the sequence, and try again. The process can take months, but the results are worth it. Right now, scientists are engineering proteins that can degrade ocean plastic. Others are creating enzymes that turn algae into biofuel. Perhaps in just a few years, we'll treat diseases not with pills, but with robotic proteins. Clean the planet, not with filters, but with artificial enzymes. And travel to Mars with proteins that transform barren Martian soil into fertile ground. Evolution spent billions of years shaping the proteins we see in nature. Now, with AI tools, we can design them in days. But what if we go even further? What if we could teach AI not just to design individual proteins, but to create entire biological systems? Think of a living cell as a computer where proteins are the programs. If that's true, then we could write a completely new operating system for life. That's the essence of synthetic biology. Scientists take an ordinary cell and replace its biological software with custom instructions. The result is a living factory that produces valuable substances on demand. For example, the company Ginkgo Bioworks has engineered yeast that produces vanillin, the same flavoring used in ice cream. It was once extracted from orchid seed pods or synthesized from petroleum. Now, microbes brew it from sugar in giant fermentation tanks. Other teams have taught yeast to make spider silk, a material 5 times stronger than steel. In nature, spiders can't be farmed because they're territorial and cannibalistic. But by inserting spider genes known as spidroins into yeast, companies like Spiber and several research groups can now grow spider silk in industrial bioreactors holding tens of thousands of liters. The protein is purified, spun into threads, and woven into fibers with strength comparable to nylon 66. These are already being tested in ballistic armor and parachute materials. Synthetic biology is also being used to fight climate change. The startup LanzaTech has created bacteria that feed on carbon monoxide and carbon dioxide, turning them into ethanol. That ethanol can then be refined into jet fuel. In the United States and China, full-scale plants are already operating, converting industrial exhaust from steel production into sustainable aviation fuel. Another idea is to create symbiotic organisms for humans, bacteria that would live in our intestines and produce all the necessary vitamins, or microbes embedded in our skin that would protect us from radiation and toxins. It sounds far-fetched, but technologically, it's possible. We already have the tools to create virtually any genetic construct. The only problem is that we don't know how to assemble them correctly. A living cell is an incredibly complex system. Thousands of chemical reactions occur simultaneously within it. They must be precisely coordinated in time and space. The slightest glitch and the cell dies. But artificial intelligence can help solve this problem. Programs have already learned to predict protein interactions. The next step is to simulate the functioning of the entire cell. A team from Stanford has created the first computer model of a living organism, the bacterium Mycoplasma genitalium. The virtual cell behaves almost exactly like a real one. It reproduces, feeds, and responds to external conditions. It's now possible to test new genetic constructs on a computer before implementing them. This speeds up the process tenfold and makes it safer. Perhaps in a decade, you yourself will be able to design new living organisms and do it as easily as you can now write computer code using AI. It could look like this. Open a genome editor, select the necessary functions from the menu, press the compile button, and get a ready-made microbe. More daring ideas concern humans. George Church's team at Harvard is working on editing the human genome, not to cure diseases, but to enhance capabilities. Imagine people who don't age, or who can see magnetic fields, or who can live on Mars without a spacesuit. It sounds like science fiction, but there are no laws of physics that prohibit it. These are engineering problems. You just need to find the right genes and insert them in the right place. Some changes have already been tested in animals. Scientists have created mice with human neurons. They turned out to be smarter than ordinary mice. Other researchers have bred pigs with organs suitable for human transplantation. Chinese scientist He Jiankui has already edited the genome of human embryos. He wanted to make the children immune to HIV. The experiment caused a scandal. He was jailed, but the technology itself works. The question isn't whether humans can be edited, but whether it should be done. Where is the line between treatment and enhancement? What if edited humans become a new race? While these questions remain open, synthetic biology is already solving more familiar problems. Pharmaceutical companies are using modified bacteria to produce insulin and other drugs. This is cheaper and safer than extracting hormones from animal tissue. In agriculture, plants with improved properties are emerging. Rice enriched with vitamin A could prevent blindness in millions of children in developing countries. Drought-resistant wheat could feed populations in the face of climate change. The food industry is also changing. Companies like Impossible Foods are creating plant-based meat that's indistinguishable from the real thing in taste and texture. And startups like Perfect Day are producing dairy proteins without cows using yeast. Environmental problems are being addressed biologically. Scientists have developed microorganisms that remove heavy metals from soil. Genetically enhanced bacteria, fungi, and algae absorb heavy metals with over 70% efficiency. There are also projects to restore coral reefs using genetically modified algae. Heat-resistant strains are being selected and developed for reef use. Aquarium tests have shown that these algae successfully support symbiosis and increase heat tolerance in sea anemones and corals. Synthetic biology is proving especially powerful in medicine. One of its most promising applications is the creation of artificial organs grown from a patient's own stem cells, a potential solution to the chronic shortage of organ donors. But the revolution goes even deeper. CAR-T therapy is a breakthrough treatment that uses a patient's own immune cells to fight cancer. Doctors extract T cells from the patient's blood and, in the lab, insert a special receptor known as a CAR a chimeric antigen receptor. This receptor trains the T cells to recognize specific proteins found on cancer cells and attack them. The modified T cells are then multiplied often into the tens of billions before being infused back into the patient through an IV. From collection to reinfusion, the process usually takes 2 to 5 weeks, but the results can be extraordinary. Unlike normal immune cells, CAR T cells don't depend on the body's usual antigen presentation system. They see their targets directly, making them less vulnerable to the tricks tumors use to hide. This precision allows them to destroy more cancer cells while minimizing harm to healthy tissue. The best results so far have been seen in blood cancers. In children with acute lymphoblastic leukemia who failed all other treatments, complete remission occurs in 80 to 90% of cases. Many adults with B-cell lymphomas also achieve lasting remissions, with some remaining cancer-free for over a decade. Side effects are rare, but possible. When large numbers of T cells attack a tumor simultaneously, the immune system can overheat, a condition called cytokine release syndrome. Symptoms include fever, low blood pressure, and, in severe cases, organ stress. Fortunately, doctors have learned to manage these reactions with medication and careful monitoring. In specialized units, the mortality rate is now below 1%. Solid tumors, however, present a tougher challenge. They release inhibitory molecules like TGF-beta, which can disarm T cells, and their dense, high-pressure tissue makes penetration difficult. As a result, outcomes aren't yet as dramatic as in blood cancers. But this doesn't mean the technology is limited, only that new barriers must be overcome. Scientists are already testing enhanced CAR T cells engineered to resist tumor suppression signals. Others can recognize multiple targets at once, a safeguard against cancers that mutate and hide their identifying proteins. Research is also advancing on regulatory T cells, which can be programmed to calm inflammation and treat autoimmune diseases. A related approach, called TCR-T therapy, trains T cells to detect internal mutations inside tumor cells, a level of personalization even deeper than CAR T. Here, scientists analyze a patient's tumor and design a receptor that fits it like a custom key. Despite its promise, this technology still faces 3 major challenges. First, time producing the cells takes weeks, and some patients simply can't wait. Second, cost a single course of therapy can reach several $100,000. Third, personalization. Each treatment is custom-made for one patient. To solve this, researchers are developing universal CAR T cells from healthy donor cells. These could be produced in advance, frozen, and delivered instantly, like a blood transfusion. If successful, this would make cell therapy accessible to millions worldwide. We are living in a time of profound transformation. Cell therapy, genetic engineering, and synthetic biology have given us the ability to intervene precisely in the body's deepest functions. These tools open the door to new cancer treatments, control of autoimmune diseases, and even regenerative medicine. But with such power comes responsibility. It is essential that these technologies remain safe, ethical, and accessible. We hold in our hands an extraordinary tool, and we must use it wisely. Naturally, this raises a question of safety. How are the long-term effects of drugs created through synthetic biology tested? After all, the technology is completely new. Once a drug is approved and released, companies continue to monitor it closely, tracking any adverse events or safety signals. When enough data accumulates for statistical analysis, researchers can draw conclusions about long-term or delayed effects, updating the drug's label if needed. In cases where a technology is entirely new yet potentially life-saving, regulators may allow its immediate use for patients with no other treatment options. In such scenarios, the potential benefits outweigh the unknown long-term risks. Is it true that CAR T therapy can cure cancers that no longer respond to chemotherapy? The answer is yes for some patients. CAR T is a truly revolutionary treatment that has shown unprecedented success against certain blood cancers. For people with advanced leukemia or lymphoma, it can bring complete remission even after all conventional treatments have failed. For many, it is nothing short of a cure. Researchers now hope to extend these results to solid tumors. But solid cancers, like those of the lung or breast, are more complex. They form dense microenvironments that block drugs and suppress immune cells. Tumors release inhibitory molecules that effectively disarm the immune system. Overcoming this barrier, breaking through the tumor's defenses, is the next great challenge. Still, progress continues. Scientists are developing smart drugs that can detect and destroy disease inside the body. Targeted drug delivery using CAR T cells, nanoparticles, or lipid membranes is already a reality. Within the next 10 to 20 years, we may see the emergence of DNA-based structures and genetic nanomachines capable of circulating through the body, detecting diseases, and repairing damage at the molecular level. Meanwhile, genome editing is opening doors that once seemed like science fiction. Using genetic scissors, known as CRISPR, scientists can now cut out faulty sections of DNA and correct mutations at their source. This technology already allows parents to protect their future children from inherited diseases, but this raises perhaps the most difficult question of all, the ethical one. How far should we go in editing a child's genome? Where is the line between preventing suffering and designing life according to personal preference? These questions demand regulation, reflection, and humility, because the power to rewrite life also means the power to redefine what it means to be human. In the coming 20 to 50 years, we will likely see new discoveries in regeneration, aging, and life extension. It's not impossible to imagine humans living 150 or even 200 years. Physicist Richard Feynman once said, "There isn't a single clue in all of biology that says death is necessary. If you want to build a perpetual motion machine, a physicist will tell you it's impossible. But there's no law in biology that says we must die. That means death is, most likely, just another disease. And one day, we'll find a way to cure it." Aging, after all, isn't directly connected to time. Time passes, yes, but aging is not inevitable. It's simply the gradual wear and tear of the materials that make up our bodies. A machine wears out too. But if its parts are constantly repaired or replaced, it can function indefinitely. The human body is no different. It's just far more complex. Imagine being 20 again. Not traveling back in time, but restoring your body to that age. That's exactly what scientists at Harvard University, led by Dr. David Sinclair, have been working on for the past 2 decades. His idea is both radical and simple. Aging is not a death sentence, it's a reading error. The problem isn't our DNA, but how it's read. Each cell in your body holds the same genetic code, but over time, the epigenome, the system that controls how that code is read, becomes damaged. It's like a scratched CD that can no longer play the right notes. The information is still there, but the body forgets how to read it correctly. The result is aging. Sinclair asked a bold question. If scratches can be removed from a CD, why can't we do the same for the epigenome? And astonishingly, he found a way. Together with his team, he activated 3 genes that are normally switched on only in embryos, genes that reset the cell's biological clock. In experiments with aging mice, this approach restored vision lost to glaucoma and age-related degeneration. The damaged optic nerves regenerated, and the old mice could see again. Then came something even more remarkable. The researchers learned to artificially age mice by inducing controlled DNA damage, causing their bodies to age rapidly. Gray fur, wrinkled skin, weakened organs. Then, using the same method, they reversed the process. The mice didn't just stop aging, they became younger. This wasn't merely a slowdown of time, it was a reversal of it. This technique builds on the work of Japanese scientist Shinya Yamanaka, who won the Nobel Prize for discovering the so-called reprogramming factors. These factors can turn adult cells back into stem cells, capable of becoming any type of tissue, even brain tissue. Sinclair demonstrated this dramatically. In his lab, his team transformed skin cells into miniature human brains, organoids, that aged like real ones. As they grew older, these tiny brains showed signs of inflammation and reduced neural activity, just like those of elderly humans. But when the researchers applied their rejuvenation method, the neurons came back to life. Electrodes recorded new bursts of electrical activity, old brain cells behaving like young ones again. Currently, the process uses viral delivery. A harmless adenovirus enters the cells and activates the rejuvenation genes. A simple antibiotic, doxycycline, acts as a switch to turn the process on. The technical details are complex, but the principle is breathtakingly simple. We are learning to control time inside the human body. For now, these results exist only in laboratory experiments, but the dream is becoming tangible. Imagine visiting your doctor once a year, receiving a short course of treatment, and watching your body rejuvenate. Sinclair believes this is not a question of if, but when. There is another way to fight aging. We are born and die, yet our genome is, in a sense, immortal. Over more than 4 billion years of evolution, there has never been a single break in the chain of life. The most ancient genes of the first living organisms are still working within our cells today. But if the genome is immortal, why does the body die? In 1972, James Watson noticed something peculiar. The protein machine that copies DNA, called DNA polymerase, cannot start reading from the very beginning of the strand. It has to attach somewhere in the middle. As a result, a few nucleotides at the end of the chromosome are left uncopied. Each time DNA is replicated, the copy becomes slightly shorter than the original. Imagine a photocopier that always skips the first and last lines of every page. With every copy, the text gets shorter. Nature found an elegant solution. At the ends of chromosomes are special, non-coding sequences, meaningless text made up of over 1,000 repeats of the same short phrase. These sequences are called telomeres. Like the plastic tips of shoelaces, telomeres protect chromosomes from fraying. But every time a cell divides, the telomeres grow shorter. After about 100 divisions, they become so short that important genes are at risk. On average, human telomeres shorten by around 30 letters per year. This is why our cells age and eventually die. By the age of 80, only about 60% of the telomere length from birth remains. So why don't we lose vital genes in reproductive cells in eggs and sperm? Because these cells produce an enzyme called telomerase, which rebuilds the chromosome ends. Telomerase was discovered in 1984 by Carol Greider and Elizabeth Blackburn. Remarkably, the same T-tag sequence appears in all vertebrates, and even in some fungi, proof that telomerase has been used since the dawn of life. Of all genes, telomerase may be the closest thing we have to the gene of youth, a true cellular elixir of immortality. When scientists introduced the telomerase gene into ordinary cells, those cells began dividing indefinitely. Their lifespan extended up to 20 times longer than normal. In most of our body's cells, however, telomerase is inactive, so telomeres shorten with each division. When they become too short, the cell's internal clock strikes its final count and division stops. This raises a deeper question. Why do we age at all? Evolutionary biologists believe the answer lies in natural selection. Evolution eliminates harmful mutations that interfere with reproduction, but it has no reason to remove genes that cause harm after reproduction. Once an organism has passed on its genes, nature no longer cares what happens next. Biologist Stephen Ostad demonstrated this beautifully by studying Virginia opossums on an island isolated from predators for 10,000 years. On the mainland, opossums live about 2 years, often killed by coyotes or owls. On the island, free from danger, they live longer and even age more slowly. Our own average telomere lifespan corresponds to 75 to 90 years, and the number of telomere repeats varies from person to person, roughly 7,000 to 10,000 at birth. This variation is hereditary, which helps explain why longevity runs in families. Studies of hundreds of centenarians from Italian villages to Japanese islands show a consistent pattern. The longest lived individuals either start with longer telomeres or lose them more slowly. Telomere length is, quite literally, a biological measure of time, a clock ticking inside every cell. A paradox lies at the heart of aging. Telomere shortening leads to cellular decline, yet without telomerase, cancer cells could not survive. Activating the telomerase gene is often the final step in a cell's transformation into a malignant one. The most famous immortal cells in history came from a woman named Henrietta Lacks in 1951. Her cervical cancer cells, known as the HeLa cell line, multiplied endlessly in the lab. They divide so aggressively that they can contaminate other cultures. When scientists block telomerase in HeLa cells, their immortality ends. They age and die after about 25 divisions. This is the essence of the paradox. The quintessence of aging is cancer. The incidence of cancer rises directly with age. In fact, aging itself is the most carcinogenic factor known. Smoking, alcohol, radiation, all these merely accelerate the process by damaging DNA and forcing cells to divide more often. Right now, as you watch this, one of your own cells has just been executed, not by age, and not by an external enemy, but by your immune system, quietly, precisely, without hesitation. That cell was on the verge of becoming cancer, and it never got the chance. These silent executions happen constantly. Every day, millions of potential traitors are caught and destroyed. Almost every cancer cell that ever arises in your body will be eliminated long before you know it existed. But this task is unimaginably complex because cancer is not an invader. It's you. It's a part of your body that has suddenly decided to act alone, even if it destroys the rest in the process. So what exactly is cancer? It's the story of cells that forget who they are. Damaged by mutations, they begin dividing uncontrollably. Cancer can arise from almost any cell, which is why there isn't one cancer but hundreds. Some grow slowly, others rapidly. Some are treatable, others relentless. Yet all share a single trait: they consume the body from within. To understand cancer, we have to look deep into the past, to the time when life consisted of single cells fighting for survival. For billions of years, cells lived alone, competing for food and space. Then, evolution discovered cooperation. Cells began to unite, to share tasks, to specialize, forming multicellular organisms. Cooperation gave them power, but it came at a cost. The individual cell was no longer the master of its fate. Its survival now served the organism as a whole. When a cell turns cancerous, it's as if it's rebelling against evolution itself, reverting to its ancient, selfish state. It stops cooperating. It takes what it needs, ignores the rules, and begins expanding without limit. It's not evil cells have no intentions, no morality. They are protein machines, following molecular instructions. When those instructions, the DNA, are damaged, behavior changes. Inside each nucleus lies your genetic code, written in 3 billion letters. Genes are recipes, instructions for building the proteins that make life possible. These instructions are transcribed, translated, and carried to ribosomes, where proteins are assembled. Those proteins determine what a cell does, how it behaves, and when it dies. But when a gene is damaged, the protein it produces is flawed. Mutations like this happen constantly, tens of thousands of times every day. Some are caused by smoking, alcohol, viruses, or ultraviolet light. But most arise simply because you're alive. DNA replication isn't perfect. Each copy is like a copy of a copy, sometimes a smudge, sometimes a tear. Most errors are harmless or quickly repaired. But over time, damage accumulates. Sometimes a break occurs in a crucial section of the genome, triggering a chain reaction. For cancer to form, usually 3 types of genes must be damaged. The first are tumor suppressor genes, the guardians that scan DNA for mistakes and stop division when something's wrong. When these fail, the cell loses control over its own growth. The second are oncogenes, genes that drive rapid growth during embryonic development, helping an organism form from a single cell in a matter of months. Once their job is done, they shut off. But if a mutation reactivates them in adulthood, the cell receives a powerful command to divide uncontrollably. It forgets that you're already grown. The third group are apoptosis genes, those responsible for programmed cell death. When a cell suffers too much damage, it activates these genes and self-destructs for the greater good. But if apoptosis genes are broken, the cell becomes immortal. It refuses to die, even when it should. And so, a young cancer cell is born. It ignores all signals. It doesn't know it's damaged. It doesn't know when to stop. It will live, grow, and consume unless it's destroyed first. That's why your immune system must find it quickly before it mutates again, before it learns to hide. Because once a cell learns to disguise itself from your immune system, it's no longer just a mutation. It's evolution happening inside you. But how can the immune system tell a good cell from a bad one? After all, they're almost identical. The secret lies in proteins. Every cell produces a specific set of proteins, and on its surface, it displays small molecular billboards that show what's happening inside. Think of them as status reports, constantly updated, revealing which proteins the cell is currently making. If a cell begins producing something strange, for instance, an abnormal protein triggered by an oncogene, the immune system notices. Specialized immune cells are trained to recognize these forbidden signals. When they see a suspicious protein displayed, they act immediately. There's no trial, no hesitation, only execution. The cell is destroyed before it can cause harm. But cancer cells are cunning. Some learn how to hide their reports, turning off the molecules that display their internal state. They erase their identity and become invisible. So what happens when a cell hides? That's when the body unleashes its other defenders, the natural killer cells. These immune warriors operate differently. They don't look for what shouldn't be there. They look for what's missing. They patrol the body, searching for cells that have gone dark. If a cell has no display case, if it hides its identity, it's presumed guilty. A natural killer doesn't ask questions, it strikes. A cell must prove it belongs. If it can't, it dies. This quiet, relentless vigilance is the reason you're still alive. Almost every cancer cell that has ever appeared in your body has been hunted down and destroyed, often before you even felt a single symptom. But this raises an obvious question. If our immune system is so powerful, why does cancer still exist? Because it's a race. Cancer evolves, mutates, and learns sometimes faster than the immune system can adapt. Eventually, a few cells escape detection. They find ways to survive under fire. That's when cancer becomes visible, when it steps out of the shadows. Yet, we evolve too. We design new drugs, train immune cells to hunt more effectively, and rewrite their genetic programs. It's not just a battle, it's an arms race between life and death, adaptation and resistance. Take lung cancer, one of modern medicine's greatest tragedies. Each year, it claims nearly 2 million lives worldwide. Around 2.5 million new cases are diagnosed annually. There are two main types: non-small cell lung cancer, which accounts for about 85% of cases, and small cell lung cancer, the more aggressive type, which makes up the remaining 15%. The tragedy lies in its silence. Lung cancer often develops quietly, without pain or visible symptoms, a master of disguise. Why? Because of where it begins. The lungs are vast, filled with millions of tiny air sacs called alveoli. A cancer cell can multiply deep within this network for years without causing trouble. The lungs keep working, oxygen levels stay normal, and because lung tissue contains almost no nerve endings, there's no pain. The tumor slowly grows from the size of a pinhead to a pea to a cherry. The person feels perfectly fine. Only when it reaches the size of a walnut or begins pressing on nearby organs do symptoms finally appear. There are, however, early warning signs. A cough that lasts more than 3 weeks without a cold, shortness of breath during ordinary activity, chest pain that worsens with deep breaths, or traces of blood in sputum, even just a few drops. These don't always mean cancer, but they always mean you should see a doctor. If lung cancer is detected early when the tumor is small and hasn't yet metastasized, the 5-year survival rate can reach 90%. But here's the problem. Only about a quarter of cases are diagnosed at stage 1. Most are discovered far too late, at stage 3 or 4. That's why doctors recommend CT scans for high-risk individuals, especially smokers and former smokers aged 50 to 80 who smoked a pack a day for at least 20 years or quit less than 15 years ago. They should undergo a low-dose CT scan once a year. Screening is also advised for people exposed to asbestos, radon, or arsenic, or those with a family history of lung cancer. Meanwhile, scientists worldwide are racing to develop new treatments. In labs, they're synthesizing molecules that can halt tumor growth, testing drug combinations that trick cancer cells into destroying themselves, and creating immunotherapies that teach the body to fight back. But the road from a discovery in a lab to a drug on a pharmacy shelf is long and difficult. It's not enough to say it seems to work. Every claim must be proven, mathematically tested, verified, analyzed. That's where the invisible but crucial work of biostatisticians begins, the mathematicians who plan and interpret clinical trials. Biostatistics is mathematics applied to human life. When a biostatistician designs a study, they decide quite literally whether thousands of people will have a chance at survival. Let's take a concrete example. A pharma company develops a drug that blocks a specific protein in lung cancer cells. Lab tests look great. Mouse studies are encouraging. Now it's time to test it in humans. But how do you do that properly? You can't just give the drug to hundreds of people and see what happens. There are too many variables: age, disease stage, prior therapies, genetics, each one capable of tilting the result. This is where the biostatistician steps in. Their job is to design a study that can answer one question with confidence: does the drug work? First comes the sample size calculation. Too few patients, and you might miss a real effect. Too many, and you waste years and a fortune. The calculation balances the expected effect size, the variability in outcomes, and the statistical power needed to detect a true difference, if it exists. Suppose the new drug is expected to increase median survival from 12 to 15 months. For families, those 3 months are priceless. Statistically, it's a small effect that can vanish inside random noise. Detecting it reliably may require thousands of participants. Patients are randomly assigned, half to the new therapy, half to standard of care. Neither doctors nor patients know who gets what double-blind randomized controlled trial. But design is only the beginning. The statistician must anticipate dropouts, handle comorbidities, specify stratification factors, for example, stage, smoking history, eGFR divided by ALK status, define primary endpoints, overall survival or progression-free survival, key secondary endpoints, response rate, quality of life, interim analyses overseen by a data safety monitoring board, and the exact timing of every lab and scan. All of this lives in a protocol, hundreds of pages of rules. Its statistical section is the blueprint for everything that follows. Then comes the hardest part, recruitment. It can take years to find people who meet the criteria, to explain the study, to obtain informed consent. Data flows in continuously. Every visit, lab, scan, and symptom is entered into an electronic system. The biostatistician and programmers watch the data for consistency, missing values, protocol deviations, and safety signals. When the study ends, the analysis begins. The question isn't just, is there a difference, but is it meaningful? Are 3 extra months worth the side effects and cost? Results are distilled into a massive report tables, graphs, sensitivity checks, intention to treat, and per protocol analyses, each number verified again and again. Because here, math isn't abstract. It decides who gets a chance to live longer. And yet, even the cleanest statistics can't solve the fundamental problem. Cancer adapts. A new drug works for a while. Then, the tumor mutates, slips around the blockade, and starts growing again. That's why researchers are pursuing more radical ideas, changing the rules of the game, so cells can't become cancerous in the first place. This is the promise of genetic engineering. Think back to childhood. If someone had told you that soon you'd buy clothes through a computer, meet people through a screen, and billions would be linked by an invisible web, you might have laughed. But here we are. Yesterday's science fiction is today's routine. Today, we find ourselves standing at another turning point, one just as strange and world-shifting as the birth of the internet. But this time, the revolution is happening inside life itself. If we truly want to understand where medicine is heading, we first need to understand where it came from and what has just changed. For thousands of years, humanity has tried to control life. We bred new varieties of wheat, tamed wolves into dogs, selected stronger horses, more fertile crops, more resilient animals. But we did all of it blindly. We didn't know why it worked, only that it did. We were sculpting life with our eyes closed. Then we found the code. DNA, deoxyribonucleic acid, a simple molecule, and yet the instruction manual for every living thing on Earth. It tells a cell how to grow, how to function, how to reproduce. All of it written in a sequence of 4 chemical letters, the nucleotides A, T, G, and C. This structure doesn't just store information, it transmits it from generation to generation. Change one letter and you can change the organism itself. That discovery turned out to be more powerful than fire or the wheel. After DNA was discovered, the age of experimentation began. In 1950s, scientists bombarded plants with radiation, hoping that random mutations would create useful traits. Sometimes it worked, but it was chaos. Progress through chance. Then came the 1970s, the birth of genetic engineering. For the first time, researchers inserted fragments of DNA from one organism into another: bacteria, plants, animals. In 1974, the first genetically modified mouse was born. These mice became indispensable tools in science and ultimately saved millions of human lives. 80s marked the commercial era. The first GMO patent went to a microbe capable of absorbing oil spills. Soon came synthetic insulin, growth hormone, and blood-clotting factors no longer extracted from animals, but produced inside living cells in the lab. And in 1994, the first genetically modified tomato, Flavor Sav, reached grocery shelves. Scientists had inserted a gene that slowed the tomato's decay, extending its shelf life by about 10 days. It was a modest improvement, but a monumental moment. Then came pigs with massive muscles, fast-growing salmon, featherless chickens, and glowing fish that you can now buy for $10 at a pet store. Humanity had learned to rewrite the script of life. But there was a problem. All of it was painfully slow. Editing DNA was expensive, complex, and imprecise. Then came CRISPR, and everything changed. CRISPR is nothing short of a biological revolution, comparable to the invention of the internet. It reduced the cost and time of genetic experiments by orders of magnitude. What once took a year can now be done in a few weeks. But where did this technology come from? The answer lies in an ancient war between bacteria and viruses. Viruses called phages are the natural predators of bacteria. Every day, they kill almost half of all bacteria in the ocean. They invade, hijack, and turn the bacterial cell into a virus factory. Most bacteria lose this battle, but some survive. And when they do, they store a fragment of the enemy's DNA inside their own genome in a molecular archive called CRISPR. When the same virus attacks again, the bacterium uses this genetic memory. It copies the stored sequence into RNA and loads it into a protein called Cas9, a molecular scalpel. Cas9 scans every piece of DNA inside the cell, searching for a perfect match. When it finds one cut, the invader's genome is sliced apart with surgical precision. Scientists realized something astonishing. Cas9 can be programmed. Give it any DNA sequence, and it will find it and cut it. We had, in essence, built a GPS for the genome. Now, we can switch genes on or off inside living cells. Any organism, from microbe to human, can be edited. In 2015, scientists used CRISPR to remove HIV from human cells in the lab. A year later, they did the same in living animals. Injecting CRISPR into infected rats reduced viral DNA in their bodies by half. It was the first proof that chronic, untreatable infections could be edited out of existence. And it didn't stop there. CRISPR began to be used against cancer. Immune cells were reprogrammed, turned into hunters capable of recognizing and killing tumors once invisible to the body. In 2016, the United States approved the first human clinical trials using CRISPR. At the same time, Chinese scientists began treating lung cancer with the same approach. In short, this is no longer a theory. It's reality. But what about congenital diseases? Today, we know that over 3,000 genetic disorders are caused by mutations in just a single gene. In many of them, changing one letter of DNA is enough to cure the disease. A classic example is sickle cell anemia. One tiny change, a single letter in the hemoglobin gene, causes red blood cells to deform into a sickle shape, clogging vessels and depriving tissues of oxygen. Scientists are now learning how to correct that single typo. A special version of Cas9 has been created that can edit one letter at a time with astonishing accuracy. But there's a problem. Conventional molecular scissors sometimes cut in the wrong place, damaging healthy genes. In medicine, such mistakes are unacceptable. That's why scientists at AstraZeneca sought a solution. They discovered a new enzyme with unprecedented precision, almost never making off-target cuts. Building on it, they created ESP-Cas9, an enhanced version of the molecular scissors, combining high accuracy with greater activity. Their results were published in Nature Communications. ESP-Cas9 preserved its perfect aim, but became far more efficient. The next challenge was delivery. How do you get these molecular tools precisely into the right cells? Researchers developed lipid nanoparticles, microscopic capsules that act like intelligent couriers, carrying gene editors directly into specific tissues. These nanoparticles can even be tailored to target particular organs or cell types. In preclinical trials, AstraZeneca scientists tested this system on the liver, targeting a gene that regulates cholesterol levels. The result was remarkable. The molecular scissors worked with surgical precision. If everything continues to progress, in the next 10 to 20 years, we could permanently cure thousands of diseases using this approach. But there's a catch. All these interventions affect only the individual. The changes are not passed on to children. To alter the course of the future, we must learn to edit embryos. And that, too, has already happened. Between 2015 and 2016, Chinese scientists conducted the first experiments with human embryos. The first attempt failed. The second was partly successful. It proved that the technology isn't perfect yet, but it's only a matter of time. What happens when thousands of genetically modified children are born? Humanity will cross a line that cannot be uncrossed. At first, parents will use the technology to cure their children of devastating diseases. Then, they'll want to improve their health, then their vision, then their intelligence. And slowly, through millions of individual choices, the entire human species will begin to change. Modified humans will become the norm, and sooner or later, someone will ask the inevitable question: if we can correct disease, can we also stop aging? Today, 2/3 of all deaths are caused by age-related diseases. We already know that aging is linked to DNA damage and the gradual breakdown of cellular repair systems. Yet there are animals in which these systems don't fail. Their DNA stays clean, their cells stay young. If we can understand their mechanisms or even borrow their genes, maybe aging isn't a destiny at all. Maybe it's just a disease. And diseases can be cured. Yes. It sounds insane, but if someone invests billions into this idea, we might become the first generation to witness the end of aging. Imagine humans who can metabolize fat effortlessly, never fall ill, and adapt to alien worlds. Humans who can travel the stars and build civilizations among them. Already, genetic testing for dozens of congenital disorders has become routine for pregnant women. And with every new possibility will come new choices and new moral questions. CRISPR is an extraordinary tool, but still imperfect. Sometimes it corrects one letter and changes an entire system. We still don't fully understand the intricate interactions between genes. We can cure one disease and accidentally create another. Precision and control, that's the next great challenge. Perhaps genetic engineering is the next step in the evolution of intelligence. A new phase where we can conquer disease, slow aging, and ultimately transcend our biological limits. The question isn't whether it will happen. The question is whether we'll be ready when it does. Yet for now, as gene editing remains confined to laboratories, we still live in the fragile bodies shaped by evolution. And the irony is many of the secrets to longevity are already within us. Over the past 2 decades, scientists have studied hundreds of thousands of centenarians around the world. The results are humbling. Our genes account for only 25% of lifespan. The remaining 75% depends on how we live. The first secret is food. In so-called blue zones regions, where people routinely live past 100 diets are simple, not strict. The residents of Okinawa follow a principle called hara hachibu, eat until you are 80% full. Research shows that calorie restriction extends lifespan in almost every species from yeast to monkeys. The reason is simple. Eating less means less oxidative damage and more efficient repair. The second secret is movement, but not the kind that comes from gyms or marathons. Long-lived people simply move often. They walk, garden, climb stairs. The key isn't intensity, it's consistency. The third secret is sleep. During deep sleep, the brain is literally washed by cerebrospinal fluid, which removes toxic proteins. Sleep deprivation accelerates Alzheimer's and shortens lifespan by years. But perhaps the most surprising factor is connection. Loneliness harms health more than smoking. People with strong relationships reduce their risk of premature death by 50%. Evolution made us social creatures. Our biology crumbles without human warmth. Another piece of advice: embrace the cold. Regular exposure to low temperatures activates brown fat, improves insulin sensitivity, and strengthens immunity. You don't need an ice bath. Even a cold shower helps. Meditation and breathing practices also matter. They've been shown to slow the shortening of telomeres, the protective caps at the ends of chromosomes. People who meditate regularly are biologically 7–10 years younger than their peers. Even the habit of learning keeps the brain young. Every new word, every skill, every challenge strengthens neural connections and protects against cognitive decline. So what do scientists recommend in simple terms? Eat 20% less than you want. Walk 10,000 steps a day. Sleep 7–8 hours in a cool, dark room. Keep your loved ones close. Fast occasionally. Take cold showers. Meditate for 10 minutes. Keep learning. These principles have worked for thousands of years, and they still work today. Genetic engineering may one day make us immortal. But for now, the secret to a long and meaningful life lies in the simple things, the ones we already have. And perhaps by the time humanity unlocks radical life extension, we'll finally be ready for it. Not just technically, but morally, because we will have learned to truly value every single day we're given.